My name is Dr. Cameron Bracken. I'm a researcher at the Centre for Cancer Biology at the University of South Australia, and I'm going to tell you about a paper that we very recently got published in a journal called Cell Systems. And it's a collaboration between ourselves at the University of South Australia, who have done a lot of the experimental so-called wet bench lab work, and researchers at the Walter and Eliza Hall Institute in Melbourne, who are computational biologists that are handling a lot of the big data that we're generating. Now, our lab is historically focused upon cancer, and specifically a process called Epithelial-Mesenchymal Transition, or EMT. And this is a process that is used in development and in wound healing, but that cancers are also able to turn on inappropriately. And when they do, that can lead to an increase in resistance to chemotherapy, and it can cause increased invasiveness of the cancer. And so it's very important to understand that process and to see whether we can target it in therapies. Now, we look specifically at the genes that control this process. And of those genes, we pay particular attention to a group of genes called microRNAs, because although microRNAs, they're small in size, they're very, very important in terms of their function because they act as master switches to control the genes downstream. What we've shown in the paper is that microRNAs can have an unexpectedly combinatorial activity functioning together to achieve an outcome which is far more effective than what they can in isolation. What the hope is in this field is that microRNAs may be useful in therapy. The problem with that, however, is that you actually need to use a tremendous amount of microRNA in a patient to achieve that effect, and that can lead to very, very deleterious consequences for that patient. The implication of our research that microRNAs that have co-evolved together can work together very, very effectively in synergy. What this provides a hope of is that perhaps if we can choose those very sort of carefully selected groups of microRNAs, then in combination they can not only affect or regulate this process in patients more effectively, but they may also be able to do it at much, much lower concentrations, and hence you'll have a more effective and a safer therapy. Although we have specifically looked at this question in breast cancer cells and EMT, we believe that the story that we have recently published is actually much more widely applicable to microRNAs generally, which is quite important because microRNAs control a wide number of processes and pathologies within the body.